Well, thanks for that really lovely introduction. It's so great to be doing something in person. I haven't talked to a live audience for 2 years, I think, and it's great to be back. And of course, it's fantastic also to see this great building. The last time I was here, I think this was just starting to be built, but it's fantastic to see it happen, and it's a wonderful place, and great to see it thriving too. So, as Jan and Richard mentioned, I've been working on DNA replication for a very, 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 very long time. And fundamentally, replication is pretty simple. We all know it from textbooks. You take parental DNA, double-stranded DNA, and each of the two strands acts as a template for the synthesis of new DNA. DNA polymerases copy the DNA, and they move in a 5' to 3' direction. So on one strand, they can synthesize continuously in the same direction that unwinding happens, and and we call that the leading strand. And the other strand is called the lagging strand, is synthesized discontinuously in these Okazaki fragments. So the two key enzymes in this whole process are the DNA polymerase, and most importantly for my talk today is the DNA helicase. So DNA helicases are the enzymes that unwind the DNA to allow the DNA to be copied. And generally, replicative DNA helicases are hexameric ATPases that encircle one single strand of DNA and use ATP hydrolysis to translocate along that strand of DNA and displace the other strand by sterically pushing it off the DNA complement. That's all fine, but if you think about it, there's an inherent biochemical problem in this, and that is that if the helicase, which is the thing that unwinds DNA, encircles single-stranded DNA, then something must happen before it can be loaded onto DNA. That is, some DNA must be unwound initially to allow the helicase to load onto the single-stranded DNA. And that's the process that we really think of as initiation of replication. And that's sort of just outlined here, that you have double-stranded DNA. Somehow you have to melt the DNA to allow the helicase to enter it. So before I kind of go into any details about proteins, I just wanted to make a point about evolution, because this is sort of different in different fields. For example, in translation, ribosomes are pretty much the same across all organisms. But that's not true in DNA replication. If you look at the three domains of life, bacteria, archaea, and eukaryotes, there was clearly a divergence at some point, very early in evolution. And the mechanisms that are involved in replicating bacterial DNA are more or less completely unrelated to the enzymology and the organization of replication of archaea and eukaryotes. The replicative helicases are completely different superfamilies. They have opposite polarities. So in the case of bacteria, the replicative helicase translocates along the lagging strand template. In eukaryotes, it's the leading strand template. The DNA polymerases are completely unrelated, and replication forks are much faster in bacteria than they are in eukaryotes. That's an inherent property of the replisome. But probably most importantly for what I'm going to talk about today is that there's an entirely different strategy used to initiate replication. So in E. coli, famously, was the Replicon model, which was proposed back in, I think, 1963 by Brenner, Cousin, and Jacob, postulated, and this really turned out to be true, that there's a single site on the E. coli chromosome. So the E. coli has a single circular chromosome. There's a single site on that chromosome. It's called REC. It acts as a replication origin, and it's at that site that the helicase gets loaded and you initiate replication. And so that site is very important in E. coli. A lot of regulation works through that sequence. But in eukaryotes, and one can even see this in archaea, which I won't go into, I'm happy to discuss later if you want, replication doesn't initiate from a single replication origin. In fact, it initiates from many, many replication origins. In typical human cell S phase, it's probably somewhere around 50,000 replication origins. And these origins can be activated. They're just drawn by these green squares. You'll see why later. So you can have some replication origins that initiate early in S phase, others that initiate late in S phase. Some of them actually never get used in a particular S phase, and we call them dormant origins, and ultimately you get the whole genome replicated. So this is great because in the case of E. coli, if E. coli wanted to add a new pathway, a new metabolic pathway, some new genes, that would come at a real cost because it would mean that the genome was bigger and it would take longer for it to replicate its genome. So that would have a direct cost to the organism. In bacteria, the time that it takes to replicate the genome is directly proportional to the genome size. That's not true in eukaryotes. Because of this multiple replication origin system, the time that it takes to replicate the genome is completely independent of genome size. It's only dependent upon the distance, to a first order approximation, between the origins, right? So if you have origins that are all very close together, you can replicate very quickly. Drosophila embryos in the first few divisions replicate their entire genomes in a couple of minutes, 1 or 2 or 3 minutes, whereas it takes E. coli 40 minutes to replicate its genome. So you can replicate a lot very quickly this way, but of course there's a price to pay, which is you've got to figure out a way to make sure that these origins fire just once in every cell cycle. So, you can't have unregulated initiation happening at these sites. They have to know that they can fire once, but they can't fire more than once. Because if you imagine one origin were to refire on a human chromosome, it would take about 3 or 4 months for those 2 forks to reach the end of the chromosome. So you really do have to regulate these origins for firing. And, over the course of many years, and many labs have contributed to this, we now have a pretty good idea. On a sort of a higher level, we understand the logic of how this process works. And it works by dividing the initiation reaction into 2 steps. So in the first step, the helicase is loaded onto DNA, and I'm going to tell you more about all of these proteins as we go along. But in the first step, the helicase is loaded onto the DNA, and in the second step, the helicase is activated. And by dividing the reaction into 2 steps and coupling these 2 steps to key cell cycle regulators, you can ensure that origins fire just once in every cell cycle. And I'll tell you how that works now. So the key regulator of replication is the famous Cyclin-Dependent Kinase. And I'm using it as a generic thing here, and I'm happy to discuss specifics about different CDKs. But for the most part, for replication, really, we just think of the cell cycle as being 2 states. A state in G1 phase when CDK activity is low. So as cells exit mitosis, CDK gets inactivated and you go into G1 phase. And then as cells enter S phase, CDK becomes reactivated. And these 2 steps are regulated differently by CDK. So on the one hand, CDKs block the helicase loading reaction. And on the other hand, they're essential for the helicase activation reaction. And so this is the logic of how you get once per cell cycle replication. When you exit mitosis and enter into G1 phase, you get rid of this inhibitor of helicase loading. And that allows you to load helicase at 50,000 replication origins, but you can't activate them at that point because you need CDK activity. Then as you go into S phase, CDKs become active, and then CDKs can catalyze the transition from helicase loading to helicase activation, but at the same time, they block the loading of helicase at origins that have already fired. And so this is really the basic logic of how you get once per cell cycle replication. There's lots of bells and whistles. It's done slightly differently in different organisms. I'm not going to go into that in too much detail. There are other kinases that are important. One of them I will mention is the DBF4 dependent kinase, which is also required for this helicase activation step. It's regulated in response to DNA damage, but I won't talk about that today. So this is just another way of looking at what I've just said. In G1 phase, CDK activities drop, allows you to load helicase, but you can't activate it, and then you go into S phase, and you can activate the helicase, but you can't load new helicase. We've spent a lot of time trying to reconstitute replication. And in fact, the discovery of this 2-step thing was really crucial for being able to reconstitute replication, because historically, people who'd work on viral systems or bacterial systems would just make an extract and throw a plasmid in, and it would replicate. But I think it was our understanding that it happens in 2 steps that really made us realize that in order to reconstitute replication, we'd have to do it in 2 different steps. So we'd have to do this first step, the helicase loading, in the absence of CDK activity, and then we'd have to do the activation in the presence of CDK activity. And I won't go through this in a lot of detail, but we knew a lot about the proteins that were involved, and there were a lot of them. We had to purify them all, and these references can show a lot of that. But so in brief, what we do is we do the MCM loading reaction first, and I'll tell you in gory detail how that works. And then we add these firing factors, including CDK, with radioactive precursor nucleotides, quench the reactions, and run them on gels. And most of I think the gels I'll show you here are all alkaline agarose gels. And this is just an early example of replication. And what you see is 2 smears of DNA. This little thing down here at the bottom is the lagging strand, and this bigger smear up here is leading strand replication. And importantly, it's dependent on everything we thought it should be dependent on. So it required ORC, which I'll show you in a minute, is involved in helicase loading. It required DDK, that kinase I just told you about. It required CDK, the other kinase I've told you about, and it required one of the key DNA polymerases. But importantly, it also had the regulation, the once per cell cycle regulation. So in this experiment, we've done what I've said at the top. We've loaded the MCMs, and then we've added S-phase CDK. But in the next line, and you see replication, but in the next line, we've added the S-phase CDK before we did the MCM loading, and I'll show you again in a few slides what that does, but it blocks replication. So this system sort of recapitulates everything we knew about replication. Okay, so what I want to do now is go through, and really what I'm going to do today is take you through what we know about how the helicase is loaded and how the helicase is activated. So the first thing to say is that the motor of the helicase is a heterohexamer of 6 MCM proteins. They're called MCM 2 to 7. They're very closely related to each other, but they're all essential. They have this conserved AAA plus ATPase domain in blue here, and they have an N-terminal collar domain shown in green here. And 5 of the 6 have these winged helix domains on the end, which I'll mention briefly. These have all got specialized roles during the helicase loading and activation steps. So I said they're all essential, and one of the reasons that they're all essential is that they all occupy a specific place in the ring. So this is the order of them in the ring, and if you don't have, for example, MCM3, MCM2 can't sit in place of it. So they all have unique places in the ring. And this also just points out what AAA plus ATPases are, how they work. Each of the subunits has an ATP binding site, which here is called a P-loop. So they each bind ATP, but then the hydrolysis is actually catalyzed by an arginine residue that comes from the adjacent subunit. So they form a sort of ATPase active site between subunits. So in budding yeast, and this is specific for budding yeast, so I'm really going to talk about how it works in budding yeast, and it's very similar in human cells, but it's slightly different. But in budding yeast, when you purify this MCM complex, you get another protein that co-purifies with it, and it's a protein called Cdt-1, which is essential for the MCM loading process. So we've done crystal structures of the Cdt-1 protein, and it's basically 2 winged helix domains, this sort of purple and this bluey color here. These are winged helix domains, and these are conserved in human cells. This green domain is specific for budding yeast, and it's related to oxygenases. It's catalytically inactive. Why it is, we don't know, but it's specific for budding yeast. What Cdt-1 does in budding yeast is something very important, and that is it binds to MCM2 and MCM6, and as you can see here, it holds the ring open between the MCM2 and 5 subunits. We often call that the MCM2-5 gate. And so in the absence of Cdt-1, the MCM complex closes, or it kind of is weakly opening and closing all the time, whereas in the presence of Cdt-1, it's held in an open configuration. So that's MCM Cdt-1. So the other key proteins involved in the MCM loading reaction are this protein called the origin recognition complex and another protein called CDC6. So ORC, origin recognition complex, is a 6 subunit protein, ORC 1 through 6. 5 of the 6 subunits are members of this AAA plus ATPase family, like the MCMs. Most of the ATPases are inactive. They've mutated key residues over time. There's only one key active site, and it's between ORC1 and ORC4, and then there's a second site that gets formed between CDC6 and ORC1. So there's these 5 AAA plus ATPases. And then the 6th ORC subunit, which I'll say more about later, isn't a AAA plus ATPase. It's its own fold that looks a bit like a TF2B. And then the CDC6 protein is another AAA plus ATPase. So I think the way to think about this is that you've got effectively a hexameric AAA plus ATPase ring between the 5 ORC subunits and CDC6. And then ORC6 is sort of sitting outside that. So ORC was originally discovered by Steve Bell in Bruce Stillman's lab as a sequence-specific DNA binding protein. And you can see one of Steve's beautiful footprints here from that original paper. You can see ORC binding very specifically at a region of a particular origin called RS1, which I'll say a lot more about today. So you can see there's protection here. And ORC binds across, so this is a sort of a linear picture of that particular origin called RS1, and there are 4 functional elements in that sequence. There's this thing called the ACS, or the A element, and then there are 3 B elements, B1, B2, and B3. This is sort of original nomenclature. But as for the moment, what's important is that B3 element is not very important. It binds a protein called ABF1. It's very important to me because it's how I got my job, but it's not actually very important for replication. But what is important is this A and B1 element. So the A element is basically essential, and the B1 element is almost essential. And these really make a bipartite binding site for the ORC protein. And you can make individual point mutations in here and knock out ORC binding. So in budding yeast, ORC is a sequence-specific DNA binding protein. But that turned out to be incredibly important for us in the field to understand how it all works, but it turns out to be a bit of an evolutionary weirdness. And the way ORC binds to DNA has been worked out by the Stillman and Joshua Tor lab and Big Ty's lab. And you can see here, I've not put any of the other structure here, but you can see there's very specifically this alpha helix here of ORC4 that sits in this major groove and binds a lot of the main contacts in that ACS element. And then there are 2 other elements, the ORC1 basic patch and a little bit of ORC2 that make specific base contacts. This is how yeast ORC binds specifically to DNA. However, when we look, for example, at that ORC4, that alpha helix in evolution, you can see that its presence is really restricted to a group of yeasts related to budding yeast, of course, and this shows it in a little bit easier to read. So in red up here, or whatever this color is, reddish color, are the yeasts that have that ORC4 alpha helix, and these all have also specific replication origins. But then as you move away from this, even within Ascomycetes, so in budding yeasts, so Candida, Pichia, Eurovia are all budding yeasts, these have now lost alpha helix, and they've lost the specific origins. When you look further away, Drosophila, plants, humans, that alpha helix is gone. So I guess the point of this is really that sequence specificity is something that arose in Ascomycetes at some point, but it's been a very useful tool, as we'll see. Okay, so the reaction basically is that ORC binds DNA, CDC6, Cdt-1, MCM get together and they load the MCM proteins onto DNA. This is the helicase loading step. Then the helicase activation step involves a whole bunch of other proteins we call firing factors, and in particular CDC45 and GINs, which get stably associated with the MCM complex to make the really active helicase. So I've told you that CDK inhibits the MCM loading process, and it does so in multiple different ways. This is just showing CDC6 levels in synchronous cells, and you can see that as soon as buds emerge in budding yeast, CDC6 disappears, reappears later in the cell cycle, and disappears again in the next cell cycle. That's because it's phosphorylated by CDK, and that phosphorylation makes binding sites for the SCF ubiquitin ligase, and so it gets degraded. So it's directly phosphorylated and inhibited by degradation. The MCM complex, and this is true for Cdt-1 as well, remember, which was closely bound to the MCM complex, is regulated by nuclear localization. This is just showing a G1 cell with the MCM complex in the nucleus and a G2 cell where it's been excluded from the nucleus. And again, this is regulated by direct phosphorylation of one of the MCM subunits on a nuclear localization export combo sequence. So these are kind of, I guess, what you call extrinsic regulatory mechanisms. So these aren't really affecting necessarily the protein's function, but other things are regulating it, so proteolysis or nuclear localization. But it turns out that ORC is actually directly regulated by CDK, and this is the explanation for the experiment I showed earlier, where CDK inhibits replication. So this is basically the same experiment as the one I showed you previously, except instead of just putting S-phase CDK in before we load MCMs, we specifically phosphorylated individual components of the MCM loading reaction and asked what happens to replication. And I think what you can see here is that it's only if you phosphorylate ORC that you block replication, and you also block MCM loading. So you can phosphorylate CDC6 or MCM in vitro, and it doesn't affect loading. So the only direct effect is on ORC. And I'll say more about that later. How do we look at MCM loading? Well, fairly simply, we have DNA with an origin on it attached to beads. We mix proteins together with various things, various cofactors, and then we can wash this away and we can look at what's left behind. And we can do this in a number of different ways: immunoblotting, silver staining, and a lot of the things I'll show you. We use the luciferase fusion at MCM3 and measure luciferase activity bound to beads. This is a silver stain example of that. So, and it makes the point that that ATP is very important in all of this. So one thing I should have mentioned already is that sequence-specific binding of ORC to DNA is dependent upon ATP. So you need to have ATP for ORC to bind DNA. But it doesn't need to be hydrolyzed. So ATP gamma S, which isn't hydrolyzed in these reactions, works as well as ATP in supporting ORC DNA binding. And so this is showing that in ATP gamma S, when we mix all of these proteins together, we get not only the ORC subunits bound to DNA, but also the 6 MCM subunits, as well as CDC6 and Cdt-1, all bound to DNA. But if we challenge that with high salt, in this case half molar sodium chloride, then all of those proteins are washed off the DNA. If we do the same experiment, but we use ATP instead of ATP gamma S, you see something fairly similar in the low salt wash. But when you do this high salt wash, now what happens is that ORC and all the cofactors are removed from the DNA, but what's left behind are the 6 MCM subunits. And these 6 MCM subunits are actually loaded into a double hexamer. So it's 2 hexamers bound in a head-to-head configuration, joined at this N-terminal collar domain. This was a low-resolution structure from Dirk Remus many years ago, and this is now, I think, probably the highest resolution structure we have at the moment. It's a cut-through so that you can see the DNA. And effectively what's happening is that this double hexamer is forming around double-stranded DNA. And if you look at the path of the DNA, you can see that it's really B-form DNA throughout the entire central channel of this double hexamer. So there's no single-stranded DNA in this at all. There's a bit of a bend in the DNA, but there's no single-stranded DNA. And remember what I told you at the very beginning is that helicases have to be bound around single-stranded DNA in order to work. So this is effectively a way of parking the MCM complex on DNA in an inactive form. Yeah, so I've told you about this already, but this sort of belies something that's actually quite interesting. So I've told you that there's this high affinity ORC binding site on the DNA, and I've told you that the MCMs are loaded as a double hexamer. This generated some interest in the field because you're effectively generating symmetry from something that's not symmetrical. So you have a single ORC binding site, but you're generating a symmetrical double hexamer on the DNA. So how does that work? There were lots of ideas about this, but I'm going to tell you how it actually works today. I've sort of gone through this already. ORC, in order to bind DNA, has to bind ATP. I haven't told you this, but you might have guessed, is that CDC6, which is the sixth AAA plus ATPase, can then bind to ORC, and that requires ATP as well. So CDC6 has to bind ATP in order to bind DNA. And ORC, when it binds DNA, generates a very sharp bend, almost 90-degree bend in the DNA, and that's still there after CDC6 is bound to DNA. So the next thing that happens is that the MCM complex interacts with this ORC-CDC6 complex on DNA, and the interaction is a very specific interaction between one of those winged helix domains on the MCM proteins, is MCM3, and CDC6 in this complex. And it's totally essential. So if this is a sequence of that C-terminal domain of MCM3, and there are multiple places we can make single point mutations in that winged helix domain and completely block MCM recruitment. And that's just shown here. This will be important in a couple of slides. So this mutant here, MCM3-11, which is a single amino acid change at the very end of the protein, completely blocks recruitment of the MCM complex in ATP gamma S and low salt. So MCM complex is first recruited via this MCM3 winged helix domain. The other winged helix domains then interact with different ORC subunits going around, and that causes a conformational change in ORC that allows the DNA to straighten out. And the configuration is such that when the DNA straightens out, it comes down into that gap between the MCM2-5 protein. And this is a very beautiful structure from Christian Speck's lab, just showing ORC in green and the MCM complex in sort of this salmon-y color. And you can see these winged helix domains here, MCM3, MCM7, MCM4, interacting with ORC, and you can see the DNA passing through the central channels of both ORC and MCM here. Oops. So then what happens is that at this point, I should say the MCM2-5 gate is still open. So the DNA is just slotted in. This is all in ATP gamma S. So this is basically that structure, what we see in ATP gamma S. It's this thing we call the OCCM complex. You can get all the way to this point without any ATP hydrolysis. So this ATP gamma S supports everything up to this point, but it won't go beyond this point. This is where you need ATP hydrolysis. And our lab and Steve Bell's lab showed a few years ago that it's actually the MCM subunits themselves that have to hydrolyze ATP in order for the ring to close, and not ORC or CDC6. So the MCM subunits hydrolyze ATP, and that causes the ring closure, and it causes expulsion of Cdt-1 from it. That's all fine. That's how you get the first hexamer loaded. But the question has been, how do you get the second hexamer loaded if you have a single ORC binding site? And the answer, to cut a long story short, is that you don't have a single ORC binding site. So the first clue that the second hexamer had to be loaded by a similar mechanism to the first came from that mutant that I showed you, that MCM3-11 mutant. So it can't be recruited at all. But if the second hexamer were loaded by a different mechanism, and one of the mechanisms that was popular at the time was that the second hexamer was recruited by virtue of interactions with the first hexamer, then that mutant should be loaded if it's mixed with wild-type protein. because the wild-type protein could be loaded as the first hexamer, and then it could be, the mutant should be able to be loaded as the second hexamer. And it turns out, I won't go through this in detail, but we showed in mixing experiments, and we've done this now in fairly sensitive luciferase assays, that you virtually never see any loading of that mutant protein when mixed with the wild-type protein in any ratio. So this really argued that the second hexamer required the same interactions as the first hexamer. And I won't go through in a lot of detail how we got to this point, but we ultimately realized that, in fact, you really do need 2 ORC binding sites to get efficient MCM loading. And an example of that shown here. So this is now a completely synthetic piece of DNA. So we've just made an optimal ORC binding site, which is the green arrow. And you can see that single ORC binding sites are very poor at MCM loading, and 2 ORC binding sites are very poor at MCM loading, unless they're in a particular orientation where they're facing each other. And then you get very efficient MCM loading. And it turns out that, in fact, the one element that I haven't told you much about is this B2 element. And it turns out that the B2 element is actually a weak ORC binding site. And so from this and from a variety of other experiments, I think it's now pretty clear that what makes a budding yeast replication origin is ORC binding sites in opposite orientations. And the typical configuration is a high affinity site flanked by multiple low affinity binding sites in the opposite orientation. We've been working on this sequence called ARS1 for about 35 years. And it's called ARS1 because it was the first one identified. And it's been analyzed to death. Every base pair, evolutionary studies have been done on it, all sorts of stuff. But it's only one of about 400 origins in budding yeast. And it turns out it's probably a little peculiar in some ways. It turns out that that B2 element is quite close to the A element. And that's not always true in yeast origins. But in the case of ARS1, it has an important implication. And that is that you can't actually get 2 ORC molecules bound to the same piece of DNA at the same time. They're sterically blocked. And so you always get it binding to the high affinity site first. This was shown many years ago by Steve Bell. This just shows that the ORC protection of the A and B1 elements. And if you make a mutation in the A element, now all of a sudden you start to see a footprint over this B2 element. And we've done it recently by band shift. And I think you can see here that with a single ORC binding site, you get a shift to this point. When you have 2 ORC binding sites in the ARS1 configuration that are about 10 base pairs apart, you mostly get a single ORC bound to it. But as soon as you move those 2 sites 2 base pairs further apart, now you can get 2 ORC bound at the same time. So ARS1 is a little peculiar in this regard. But it turned out to be useful because it allowed us to, again, dissect multiple steps. Okay, so the problem here is we've loaded the first hexamer. We know that that B2 element is going to be important for ORC binding, but the problem is that the B2 element is underneath the MCM hexamer that's been loaded. So what has to happen now is that somehow ORC has to release from DNA, and this, has to be released from DNA. And the way we think this happens, and I have to say this is still a slightly weak point in the model that doesn't have a lot of experimental evidence for it. Remember I told you ORC needs to be able to bind ATP in order to bind to its specific sequence. ADP doesn't support sequence-specific binding. So if ORC were to hydrolyze ATP to ADP, it presumably would lose its affinity for DNA. And that's what we think happens. And one of the reasons for thinking that is shown here is that if we just take that winged helix domain of MCM3 and we add it to ORC CDC6, it greatly stimulates the ATPase activity of ORC. So we think that something in these first steps is actually triggering ATP hydrolysis by ORC, which is then allowing ORC to be released from the DNA. Once it's released from DNA, then the MCM is free to slide because it's only topologically bound around the DNA. So that presumably is what allows ORC to bind to that B2 element. And in fact, we were able to capture this together with my colleague Alessandro Costa, and a lot of the structures I'll show from here on in are with Alessandro, who's been a great collaborator. We were able to capture a novel intermediate in the MCM loading reaction, which we call MO for MCM ORC. And it shows MCM bound on the other side of that loaded single hexamer. So instead of making contacts with the C terminus, which is what's happening here, in this complex, ORC is now bound to that B2 element. It's bent the DNA, and it's bound to the N terminus of the MCM complex. And this structure had a lot of implications because the way that this is configured, this has bent the DNA very similar to what's happening over here. And this face of ORC is now free to bind the second hexamer. And we know we've seen this happen. So if we take this structure and we add more MCM to it, we can see that second hexamer being recruited. And again, you can see the DNA running through the center of this ORC MCM complex. So we think that that's how the second hexamer gets recruited. It's by the same mechanism that the first hexamer is recruited. And then presumably, and again, this is a step that hasn't been completely worked out, or can release this first hexamer and load the second hexamer, and then you get your double hexamer formed. In this model, this second ORC can be the same ORC molecule, or it can be a new ORC molecule. And we think that whether it's the same molecule or a different one is dependent on ORC concentration. So at low ORC concentration, there's a way that ORC can flip over to bind to this second element, which Steve Bell has recently shown in some nice FRET experiments. But at higher ORC concentration, it's probably almost always a second ORC molecule. One of the things that I haven't talked about, but I've sort of inferred, is that the distance between the ORC binding sites isn't terribly important for MCM loading. So it can work when even the two ORC molecules, ORC binding sites overlap, but you can actually get very efficient collaboration of two ORC binding sites, even when the sites are 400 base pairs apart. So anywhere from 25 to 400 base pairs apart, and you get efficient MCM loading. There's clearly a peak at around 70 or 80 base pairs, which I'll come back to. That is more or less the sweet spot of where the size of a double hexamer is about 70 base pairs. And I should say that all of these synthetic sequences are functional replication origins in vivo, efficient functional replication origins. So it's not a kind of an artifact. So what I've sort of implied is that there's multiple ways that MCMs can be loaded. So there's this way that we've learned from RS1, which is very sequential. ORC has to bind to one site, has to load an MCM, it has to release. You have to get a second ORC binding event and all of that. But actually, the flexible distance requirements mean that there's other ways that the MCM complex can be loaded. So for example, when you have two sites that are far apart, presumably each can bind and load an MCM complex, and then somehow they have to translocate to meet each other. And we know that if we put a roadblock between two ORC binding sites, it completely blocks double hexamer formation. And then there's sort of the third kind of way it might work, which is when the two sites are at an optimum distance apart. And this turned out to be quite interesting to us because of this thing that I've shown you already, that ORC is inhibited by CDK phosphorylation. So we started to look in a little more detail into how CDK inhibits ORC's MCM loading ability. And we knew two of the subunits of ORC, ORC2 and ORC6, have CDK phosphorylation sites, and we can make mutants that block phosphorylation. So I'll just show here, you can see, for example, ORC2 gets shifted when you phosphorylate it, but in the ORC2 mutant, you don't see that shift, and similar for ORC6. We can now start to ask, what is it that phosphorylation blocks in MCM loading? As you can see here, whether we phosphorylate the entire ORC complex, we just phosphorylate ORC6, or we just phosphorylate ORC2, we pretty much block double hexamer formation. This is by EM. If we look at what happens to that first step, the OCCM, just the intermediate before ATP hydrolysis happens in ATP gamma S, we can see that CDK phosphorylation probably inhibits about 50%. It reduces OCCM formation. It doesn't actually kill it. And that seems to be entirely due to ORC6 phosphorylation, because ORC6 phosphorylation does pretty much the same thing, whereas ORC2 phosphorylation doesn't affect OCCM formation at all. But I think the most important thing is the formation of that MO complex. So it turns out that phosphorylation of either ORC2 or ORC6 completely blocks the formation of MO. It's that step that's being blocked by CDK phosphorylation, and that's going to be important in the next part of the talk. That wasn't so surprising to us because this is ORC6. It's between ORC and the MCM complex in this MO formation, and the N-terminus of ORC2 also runs into the MCM complex up here. It's a slight digression, but it's sort of how we got to understand this a little bit more. Many years ago, Joachim Lee's lab did some experiments where they were looking at the role of all these different pathways in preventing re-replication. And what they found was that if they built a cell that was entirely dependent upon ORC phosphorylation to block re-replication, so they deregulated the CDC6 pathway, deregulate the MCM pathway, now it's only ORC that's blocking re-replication. Then there was actually a small number of origins that seemed to refire in that particular condition. So there's a subset of origins. The best of these is this origin called RS317 that are not inhibited efficiently by CDK phosphorylation of ORC. But they did some analysis of the sequence, and they found that like most yeast replication origins, it had this sequence relatively close to the high affinity site, this green site, that was important for just generic origin function. So this has the B2-like elements in it. If you look at the sequence, it's got lots of near matches to the ORC binding site. But then they identified this thing they called a reinitiation promoter that was further away. And this was absolutely essential for reinitiation, but wasn't required for normal replication. So we've now taken RS317 and asked, what does CDK do to loading? And you can see here, this is what CDK does to loading at RS1. It completely blocks it. And when you look at RS317, you can see that there's quite a bit of residual MCM loading activity that occurs even with the phosphorylated ORC. So this is presumably reflecting what's happening in vivo. And nicely, if we remove that reinitiation promoter, now the loading drops down to the level that we see with RS1. Now, it turns out that reinitiation promoter, when we started to look at it, has multiple weak ORC binding sites in it. And we can show that ORC binds to these sequences just like it binds to the B2 element. So this is looking at ORC binding to that replication initiation promoter, and you can see it binds as well as it binds to B2. Like B2, that binding is salt sensitive compared to the high affinity sites, but it binds pretty efficiently to it. And that made us begin to wonder whether the spacing of the ORC binding sites might be important in this CDK regulation. And quite amazingly, when we went back to that sequence of different ORC binding sites and their distances, what we found was that actually when the ORC binding sites are 90-ish base pairs apart, we get almost as efficient loading with CDK phosphorylated ORC as we see with unphosphorylated ORC. So at that sort of sweet spot of 90 base pairs apart, CDK phosphorylation doesn't block MCM loading. And remember that phosphorylation blocks the formation of that MO complex, that MO intermediate. So what this is, I think, telling us, I should say that this 90 base pair sequence re-replicates really efficiently in vitro and in vivo. I'll skip that. But I think the important thing is that CDK phosphorylated ORC can't form this MO complex. And so the implication is that at this spacing of ORC binding sites, you don't need the MO formation. And so I think this is telling us this and lots of other things, is that actually the loading of the double hexamer is quite a flexible pathway. You can have this sequential loading, which is what I showed you for RS1. You can have this 90 base pair MO independent mechanism, which I've shown you, and you can probably, we haven't investigated this as much, but there's probably some kind of a pathway when the sites are far apart that involves translocation of at least one of the MCM hexamers on DNA. So I just wanted to sort of end this section with a little evolutionary kind of a comment, which is that in archaea, which I told you is sort of on the same evolutionary pathway of replication as eukaryotes, replication origins actually have this configuration. They have two high affinity binding sites separated by about 70 or 80 base pairs. Budding yeast, we think, has re-evolved something similar to this, where it has high affinity sites and low affinity sites. And vertebrates are still a bit of a mystery. Their ORC is a non-specific DNA binding protein, but we postulate is that it will work in a similar mechanism, but using lots of low affinity binding sites. So I guess rather than thinking about MCM loading as a kind of a dance with very specific regimented steps, it's probably better to think of it as a kind of a more freeform jitterbug type dance. I would just go through a little bit about how the helicase gets activated. So I've told you already that when the MCMs are loaded onto DNA, they hydrolyze ATP in order to make the double hexamer. And once they're hydrolyzed ATP, they actually remain stably bound to ADP. And then what happens is that those firing factors in the activation step get recruited to the double hexamer, and that triggers the release of ADP. And that becomes important in a minute. So how does that complex form? Well, this is where the phosphorylation is critical. So the DBF4-dependent kinase phosphorylates the MCM subunits, and that generates binding sites for this SLD3 protein. And SLD3 then recruits CDC45, and CDC45 is one of the essential components of the CMG helicase. CDK then phosphorylates SLD3 and SLD2, another protein, and they get pulled together by this multiple BRCT repeat protein, DPB11. And SLD2 pulls in DNA polymerase epsilon, which is essential for CMG formation, but it also recruits in the GINS complex, which is the other part of CMG. That leads to ATP release. And then it's the rebinding of ATP that triggers the next key conformational change. This doesn't require ATP hydrolysis, but the rebinding of ATP causes the stable association of CDC45 and GINS in a CMG complex, and the first melting of the DNA. And I've overrun myself quite a bit, and I'm going to do disservice to this great structure that's been generated by Jacob Lewis in Alessandro Costa's lab, but he's now been able to generate a high-resolution cryo-EM structure of that double CMG complex with the melted DNA in the middle of it. And this is it. This is the MCM ring, CDC45 and GINS, and DNA polymerase epsilon. You can see it in all its glory here, and you probably can already see that the MCM subunits have really rotated quite a lot relative to where they were in the double hexamer, and that's just shown in this little animation. So the MCM subunits have really turned apart quite a bit. There's lots of different contact changes, which I won't go into, but I think the key thing I wanted to get across is that we can now see the first evidence of DNA melting in the DNA in the core. And there are these blue bases here and here in each of the two hexamers. And again, I've gone through this too quickly, but there's a key to this is a small domain in MCM2, one of the MCM subunits, which inserts into the minor groove of the DNA and causes this untwisting, which you can see here. Yeah, so you can see that domain inserting into the minor groove. We've mutated some residues in that domain. They don't block MCM loading. They don't block CMG formation. They completely kill replication, and they completely kill the melting, which we can see using a topological assay that I should have gone through, but I didn't. We understand pretty much everything up to this point where we get this melted intermediate. What happens next is still a bit of a mystery. You need ATP hydrolysis for the next step. You need another protein called MCM10. Somehow that leads to the extrusion of the lagging strand template and the reclosing of the ring around the leading strand template. That's something we need to understand next. And then what happens is that the two active helicases are both on the leading strands of leading strand templates, and they actually pass each other then in the origin. Given the time, I'll stop there. And I really just want to acknowledge some of the people who were involved in this. There's lots of ex-lab members. In terms of current lab members, Chu Thang was really important in a lot of the MCM loading stuff that I talked about. Marta did a lot of work with Jacob Lewis on that structure at the end. And again, I have to thank my collaborator, Alessandra Costa, who's been absolutely amazing. We've had a wonderful time doing a lot of this stuff, and all the people in his lab who've been involved in this. So I'll just stop there and take questions. Thank you very much.